Okay, so magandang araw sa ating lahat. Last time, natapos na natin ang discussion natin with carbohydrates. So now, we'll move on to proteins and all the related topics such as amino acids and peptides. So naturally, ang unang tanong natin is, ano nga ba yung proteins? So ito yung pinaka-abundant na biological macromolecules dahil nag-occur ito sa lahat ng cells at sa lahat ng parts ng cells. Nag-occur din siya in great variety. May thousands of different kinds at meron ding iba-ibang sizes ranging from relatively small peptides to huge polymers with molecular weights in the millions. Nag-exhibit din siya ng enormous diversity ng biological function and are the most important final products ng information pathways. Ang proteins din ay molecular instruments through which yung genetic information natin ay na-express. Pwede rin siya ma-break down into their constituent amino acids by various methods. So for today, i-discuss natin yung amino acids, peptides, and naturally yung protein. So bakit natin sinala yung amino acids? That is because yung proteins natin are polymers of amino acids, with each amino acid residue joined to its neighbor by a specific type of covalent bond. So yung amino acid natin is a compound na naglalaman ng amino group, which can be seen sa illustration, and a carboxyl group. Meron din tayong alpha group and R group. So as I said earlier, ang ating alpha amino acid ay may amino group na naka-attach sa carbon adjacent sa ating carboxyl group. And yung alpha carbon sa ating alpha amino acid is also bound to a side chain group, which is called R. Yung R natin may contain different elements because R gives the identity to the amino acid. And only 20 amino acids are usually found in proteins. So, meron tayong 3-letter or 1-letter code and nire-refer natin ang ating proteins by that. We can classify amino acids by their pH and their charge. So, yung non-polar natin na amino acids include glycine, which have hydrogen as its R group, and then alanine na naglalaman ng methyl as its R group, then valine, cysteine na may sulfur, proline na cyclic ang structure, and leucine, isoleucine, methionine na meron ding sulfur, tryptophan, and phenylalanine. While yung positively charged naman natin na amino acids include glycine and arginine with long carbon chains, and then histidine. Then we also have our polar amino acids, which include serine, threonine, tyrosine, asparagine, and glutamine. While yung negatively charged amino acids natin include yung aspartic acid and yung glutamic acid. Nakaklasify din natin yung amino acids natin as non-essential. Ito yung mga amino acids na nagagawa ng ating katawan and can be gotten from food. So, example would be alanine, asparagine, aspartic, and glutamic acid, as well as serine. Yung conditionally essential amino acids naman natin ay napaproduce ng mga malulusog na katawan, pero hindi siya pwedeng maproduce kapag gutom ka or starving or meron kang metabolism error. These are arginine, cysteine, glutamine, glycine, proline, and tyrosine. Well, the last one are essential amino acids. These essential amino acids are only gotten through food. So, hindi mo siya makukuha sa iba pa at hindi din siya readily available sa katawan. These include histidine, isoleucine, leucine, valine, methionine, phenylalanine, threonine, tryptophan, and our glycine. May mga reactions din sa ating amino acid na nagpo-form ng peptides. Ito yung peptides natin ay na-form through peptide bond. So, ang isang peptide bond is a chemical bond formed between two molecules when yung carboxyl group ng isang molecule ay nag-react sa amino group nung isa pa ng molecule na magre-release ng water. Ang tawag dito is dehydration synthesis reaction or tinatawag din natin condensation reaction. At usually, nag-occur siya between amino acids. So, more on the structure of our peptides. Yung peptides natin are a series of amino acids like sa illustration natin below. Like yung R1, R2, R3, R4, R5, and R6 ay nagre-represent ng iba-ibang amino acids. So, we have both terminals there, which are called amino terminal dahil ito ay nagka-contain ng amino group. Pwede rin natin siyang tawaging N-terminal. And the other one is the carboxy terminal or what we call C-terminal because nagka-contain siya ng ating carboxyl group. Now, we'll go to peptides. So, ano yung peptide? It's a short polymer ng amino acid joined by peptide bonds. Ito ay classified then by the number of amino acids sa chain. So, some vocabulary words would include dipeptide, which is a molecule containing 2 amino acids joined by a peptide bond, hence di. While we have tripeptide, so from the word tri, molecule containing 3 amino acids joined by peptide bonds. While we have polypeptide. Yung polypeptide natin is macromolecule na siya containing many amino acids joined by peptide bonds. So now, finally, we'll go to our proteins. Yung proteins natin ay nahahanap sa mga pagkain such as eggs, dairy, seafood, legumes, meat, nuts, and seeds. Yung proteins natin ay tumutulong sa paglaban mula sa infections, nakakatulong din siya sa cell division, sa information, sa pag-maintain at pag-construct ng ating living cells. Yung biological macromolecule na may molecular weight na 5,000 grams per mole o higit pa ay nagkakonsist ng 1 or more polypeptide chain ay tinatawag nating protein. Protein is also coded by genes. Proteins can be storage, structural, transport, antibodies, pwede rin siyang catalyst, tinatawag natin silang enzymes, and regulatory proteins, like for example, hormones. Since ang proteins natin ay naglalaman ng maraming amino acids na bonded through peptide bonds, maraming conformations ang possible sa proteins due to the flexibility ng amino acids natin. So, yung change sa isang amino acid sa sequence can alter biological function. Example, hemoglobin associated with sickle cell anemia. So, yung protein structure natin ay may levels na yan because of the number of amino acids here. So, meron tayong primary protein structure. So, ito yung sequence ng amino acids sa isang polypeptide chain. So, as I said earlier, meron tayong terminal, the N and the C-terminal end. Binabasa natin yung primary protein structure natin from the N-terminal or the amino group terminal to the C-terminal end or sa carboxyl terminal end natin. And then, we have our secondary structure. Ito na yung ordered arrangement or conformations sa localized regions ng ating polypeptide chain. Ito yung nagre-refer siya sa interactions ng peptide backbone natin. Example, alpha helix or beta pleated sheet. So, types ng secondary structure natin is ang alpha helix. Coil siya ng helix which is clockwise or right-handed. At lahat ng R groups natin ay nagpo-point outward from the helix. While yung beta sheet are made of beta strands which are extended stretches ng 5 o higit pang amino acid na naka-organize na magkakatabi or next to each other. So, we also have our bender loop, polypeptide chains na pwedeng mag-fold upon themselves na mag-perform ng bend or loop. So, yung bends naman, usually, nasa surface siya ng globular proteins. And then we have our tertiary structure. So, it can also be called 3D arrangement of all atoms. Like in the illustration, naglalaman ng beta sheets or alpha helices yung ating tertiary protein structure. So, ano yung forces na nag-drive sa tertiary structure natin? Ito ay non-covalent interactions such as hydrogen bonding between yung sa side chains. And then we have hydrophobic interactions as well as electrostatic attractions, electrostatic repulsions, and then we have our covalent interaction. Meron tayong classes of proteins depending on their 3D structure or tertiary structure nila. We have our fibrous proteins. Yung fibrous proteins natin, nagko-contain ng polypeptide chains na organized approximately parallel along a single axis, like what is described in our illustration. Ito yung nagko-consist ng long fibers or large sheets. And mechanically strong din to. Insoluble siya sa tubig and dilute salt solutions. Also, nagpe-play siya ng important structural roles in nature. An example would be keratin and collagen sa ating connective tissues such as cartilage, bones, teeth, skin, and blood vessels. Then next natin is globular proteins. Ito yung proteins na folded sa more or less spherical shape, hence the name globular. They tend to be soluble sa tubig at sa salt solutions. Most ng polar side chains nila ay nasa labas and can interact with the aqueous environment by hydrogen bonding and ion dipole interaction. So we can call this polar side chains as hydrophilic or water-loving because they can interact with water. While most naman sa non-polar side chains nila are nasa loob lang, buried inside. We can also call this as the hydrophobic side. Nearly all have substantial reactions of alpha helix and beta sheet. So now we'll go to breaking down proteins. We can break down proteins through denaturation. So in denaturation natin is the loss of the structural order, like the secondary, tertiary, quaternary, or combination itong lahat na nagre-result sa loss ng biological activity ng ating protein. Pwede natin siyang gawin via heat or via ultraviolet or UV or through strong acids and bases. Pwede tayong gumamit ng detergents such as sodium dodecyl sulfate or yung SDS na nagdi-disrupt ng hydrophobic interactions natin. Pwede rin natin gamitin yung urea or guanidine na nagdi-disrupt naman ng hydrogen bonding. Pwede rin tayong gumamit ng reducing agent like mercaptoethanol na nagre-reduce ng disulfide bonds. Pwede rin tayong gumamit ng organic solvents, example, ethanol and acetone, because yung change sa dielectric constant and hydration ng ionic groups ay mag-interfere with intramolecular hydrogen bonds. And then we can also denature proteins by agitation or shearing of hydrogen bonds. Like for example, pagbati ng itlog or beating the egg white albumin into meringue. Lastly, we have our quaternary structure. Ito na yung arrangement ng monomer subunits or ng polypeptides with respect to each other. So yung quaternary protein natin is a protein consisting of more than one amino acid chain. So lahat-lahat na ng biggest picture na ng mula sa ating primary, secondary, and tertiary protein structure.